This is a special episode of the Immunology Podcast, Immunology 2023, Day 3. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rad. Welcome back to the Immunology Podcast. We're having conversations with immunologists. We're back with our third episode covering all the highlights of Immunology 2023 here in Washington, D.C. Brenda and I have had a bunch of fun attending and getting to meet you all, but it's not over yet. If you're looking to find out what we're up to at the podcast, follow us on Twitter at IMMUNOPodcast. You can find episodes covering all of the 2 previous days of the meeting at www.immunologypodcast.com or wherever you find your podcasts. Today we'll again be discussing some of our favorite sessions throughout day 3 of the meeting, so if you were in another session and weren't able to attend the meeting, we've got you covered. We'll be kicking things off in just a minute, but before we get to that,
whether you're looking to attend an immunology conference this year or to expand your network, make the most of your experience by downloading STEMCELL Technologies' collection of tools to help you prepare for your next event. The downloadable checklist and guides include recommendations on how to get ready before attending conferences, tips for networking, best practices for your LinkedIn profile, and more. Download the conference toolkit at www.stemcell.com/conference-toolkit.
all right, Brenda, it's day 3.
Day 3. Exciting. Man, so much science, so little time.
I know.
How are you holding on?
I'm okay.
Yeah.
I'm a little deep in the conference mode. I'm not gonna lie.
Yeah, me too.
Well, we're recording at the end of day 3. That means tomorrow's Mother's Day, and flowers are already delivered to my wife and my mother.
That's so nice. Very well. I'm going to wish my mom happy birthday. And happy birthday, happy Mother's Day. But you know what also? It's exactly 33 years to the point she became a mom. It's my birthday tomorrow.
I know.
So what did you get me, Jason?
You have to wait until tomorrow, but I actually planned something.
Really? Oh my God. Dear listeners, I'm very excited. Well, I'll tell you tomorrow what I got for my birthday.
I couldn't get everyone at the conference to sing happy birthday. Oh, that would be amazing.
Wouldn't that be nice? Well, maybe some other time.
Or at least a symposia. But speaking of symposia, what did you watch, Brenda, here today?
Well, first of all, I just want to say that yesterday I went to Doug Green's presentation. He was a guest at the podcast, and I really like his story. So one of the first story he shared, which I had been following before, but I think is super cool. He talks about the importance of spatial kind of localization of proteins and transcription factors in activated T cells. And he shows that particularly for the case of MYC, with MYC, of course, as any protein gets translated in the cytoplasm, because it's a transcription factor, it needs to be shipped into the nucleus to act. But then as T cells are activating this, so they may make a synapsis with a DC presenting an antigen, and then this synapsis generates a whole change of the cytoskeleton of the cell and a polarization to the synapses, and a bunch of this MYC gets kind of brought to that side of the cell. So you end up with a situation in which a CD8 cell ends up having one side that is on the synapse that is kind of higher on MYC, and then he also saw he's higher on CD8 co-receptor, and then the other side has less. And this actually, once the cell divides, this imbalance stays, and then you end up with 2 daughter cells. One of them has more CD8 and more MYC, and the other one has less. And MYC is associated with effector function and proliferation, and so it has a lot of downstream effects that are associated with effector T cells. And then what he shows is that the cell that has the MYC has a tendency to become an effector cell, whereas the other cell that gets the less of the MYC is more prone to becoming a memory cell. And this is because of this, at least in part because of this distribution, this physical distribution of proteins when the division of the cell happens. So I thought it was really cool. I had already kind of knew that story. He talked about it, was very articulate about it. And I think a lot of people were very interested. So kudos to Douglas Green. It was really nice to also have him in the podcast. So that was yesterday, though. And also yesterday, there was a very special guest, Anthony Fauci. For those who live inside a box, the former director of the
NIAID.
And of course, the face of the COVID response here in the U.S.
Yep.
So how was the talk?
It was good. He went over kind of lessons learned from the pandemic from a very biological perspective. As others said, I don't think anything was really surprising, but putting it all together was interesting to see. And so I thought it was well done. It was also nice to see a bunch of scientists, having a little fan experience with the chief scientist of their own field who had such a public job recently. So it's much different than usually what you get politically.
Okay, so which talks did you go today?
So I mostly did major symposia C on inflammation and injury and other fun things, and some innate receptors. So we had Ruslan Menzatov, very famous immunologist, talking about type 1 versus type 2 inflammation after injury. And he was talking about in a model system with his last B protein, which comes, I believe, from Pseudomonas, that if you get a high dose of it, that prompts a type 1 response. But if you get a low dose of it, it prompts a type 2. And then interestingly, if you get repeat exposure to type 1, it's protective. So repeat high dose, that has that protective effect. You develop tolerance, you respond better to damage on the second go around. But that if you do repeat type 2, that's allergy. And so he's trying to understand some of the mechanisms that are driving this. And at least it appears that he hasn't figured out the dose-dependent effect, but either way, STAT6, it's driven by a STAT6 system, because STAT6 knockout mice are protected, like they have last B exposure, and similarly, damage does not require an adaptive immune system. So what he was able to show as well is that sometimes the tissue damage is working through a heme recognition system by the NRF2 pathway. So when you have tissue damage, you have blood lysis that releases heme. And so there's this IL-4/13 STAT6 pathway is an antagonist to the heme pathway. And so that's why STAT6 knockout have better responses. They're going to sense the heme better. And so if you sense heme, there's an injury, your body tries to figure out what to do about it, repair itself, develop protective effects. So that was interesting to know that injury was running through a heme sensor. Makes sense. You know you're bleeding because you look at it and go, I'm bleeding. Your body does the same thing. I like the parallels that way. Next up was Luis Barrio. He was looking at genetic and epigenetic determinations of inter-individual variation in innate immune responses. So he's a geneticist. And long and short, he was really looking at the fact that 60,000 years ago, a group of humans left Africa, and we're all descended, if you're not of African descent, we're all descended from that same group, which means there's a genetic I don't want to say break, but a genetic drift that you can track from that event, from people moving to new antigen exposure. And so they found that there is, in African ancestry, a stronger inflammatory response towards, generally speaking, and especially towards Listeria and Salmonella. And so there's been an adaptation in people of European ancestry against those that have less inflammation. And interestingly, he also talked about the Neanderthal thing, which is this Neanderthal haplotype gene. Human Homo sapiens encountered some Neanderthals, and there was some breeding that occurred. And we all have a little Neanderthal DNA in us if we're from European ancestry. How it got there, ask grandma.
Great grandma, most likely.
Yes. But there's a specific gene, OAS1, that has a survivorship bias against Yersinia, the Black Plague, and it protects against COVID-19 susceptibility and severity. And there's also epigenetic differences as well. So that's a fun one, just weird, nerdy, evolutionary immunology. Tim Sullivan talked about transcription epigenetic regulation of NK cells. So from here, really, he went into a couple different things in some older papers that he kind of talked about. But what I thought was really fun was that he looked at man flu. So if you're familiar with man flu is the notion that men, when they get sick, whine and complain more about how much it aches. But it actually has to do with sexual dimorphism and cytokine production and activity in immune cells. And so apparently men have more NK cells, but each NK cell produces less interferon gamma. But on net, you have more interferon gamma from the total extra immune cells. So it's not hormonally driven, and so that means it has to be genetically driven. And obviously women, they have 2 copies of everything because the Y chromosome is just defective. But to make sure there's chromosome or genetic balance, women undergo selective X-linked silencing. But some genes stay on no matter what. So not everything gets silenced. And so he looked at what was conserved And this is a gene called UTX, conserved X-linked escapee is a gene called UTX. It binds and demethylates DNA, and using a cut and tag approach, because you can't get a lot of NK cells, so it's different than just a chromosome mapping you typically do, like a CyTOF or something, right? That. So instead, they did this system where you could see where it binds, you cut it, and then you see that tagged region in sequence, so you can see where it's tagging. And lo and behold, it binds the interferon gamma promoter, and its absence has decreased accessibility to the genome for interferon gamma, which is why each of a male's NK cells produce less interferon gamma. Thank you very much, genetics. And then we had Ai-Ling Lim. She looked at pre-birth immune education, just starting her own lab. And in that one, she was able to look at this notion that transient maternal infection elevates Th17 accumulation only in the gastrointestinal tract of offspring, and it happens during weaning and persists into adulthood. Doesn't matter who delivers the baby, right? It happens what uterus the mouse is in. So if you take an infected mommy and then do an embryo transfer into that after the infection, that still passes the phenotype. And they found that it's a serum soluble factor is enough to transfer the effect. So if you just take the blood from a mommy that was as and put in the other mommy that has the baby in it, right? So infected female and then transfer it over, it will transfer. It's IL-6 and microbiota dependent, and maternal IL-6 exposure alters the offspring's intestinal milieu and promotes a tonic response to the microbiota through the fetal gut having an IL-6 STAT3 axis. Now, it enhances survival with infection, but makes outcomes of chemical colitis and wound repair worse. And then in this talk, we saw one of our guests, Shruti Natick, came on and talked again, talking about IL-17 and skin organoids, talked about her model of the basically recapitulated psoriasis in a skin organoid system. And they found that what was really cool is that if you trigger a cell with IL-17, these organoids, they have a second response that persists higher, so a tonic increase in activity. You can take the cells, you can passage them, split them up, mix them up, flip them around, reverse it, do the whole Missy Elliott thing on them, and they still maintain this effect after passaging. But they looked at human, because they developed these skin models from actually foreskin discards from circumcisions. So you get human genetic variants, and they find the amount of this is variable. So there's some either genetic or maternal imprinting, like we just discussed as well. And then lastly, I went to one of my old lab colleagues' talks on T-cell migration, and she presented her work. It was actually a chunk of my work from back in the past that's finally being presented on a family protein called TIP proteins, which are proteins that regulate phospholipid signaling by binding and holding PIP2 in the cytoplasm of cells. And then they can control the leading and trailing edge during chemotaxis by regulating the PI3K PIP3 wave. And so it has some pretty novel biology there that she was talking about. So that was my day.
Very cool. I really like those talks. So very nice. I also went to a bunch of talks, and I first started. So I did also a little bit of session hopping, and I started with the SITC symposium, the Society for the Immunotherapy of Cancer, and I attended the talk of Lisa Butterfield from University of California, San Francisco, in which she was looking at dendritic cell dysfunction and what things we can learn to make better vaccines. Like he was basically talking about the idea that autologous DC vaccination, that is when you, for example, you have dendritic cells and you load them with an antigen or a lysate, and so on, you expand them and you bring that to the patient to try to educate the immune system, has really shown some success. Especially there are some recent clinical trials with from some solid cancers that have shown a level of protection or like therapeutic efficaciousness. And so, but she was looking into why some of them don't work, and why also when you get dendritic cells from actual patients, they're also usually less efficacious than dendritic cells derived from monocytes of healthy people, of healthy donors. Basically, kind of long story short, she did vaccinations with monocyte derived dendritic cells that were induced within GM-CSF, and she loaded the dendritic cells with antigen using adenoviral transduction, and she showed that, in such a system, these dendritic cells are educating CD8 cells. They become, which are critical for their response, and she followed these responses, and she sees that sometimes this dendritic cells are not really working properly, and she looks like there's one of the main issues with kind of faulty dendritic cells has to do with NF-kappa B signaling and the correct translocation of NF-kappa B into the nucleus of the dendritic cells. This results in reduced ICOS ligand expression, and this correlates with lower T cell responses of CD8 T cells in vivo. And she did this couple of assays in which she was looking into metabolism of these dendritic cells. She mentioned an assay called CNEF, which is based a cytometry-based assay to look into metabolic markers and metabolic function of cells, and also mass cytometry, in which is profiling metabolic enzymes and transporters and receptors, and try to use that to characterize the metabolic status of the cells. And she shows that these dendritic cells that seem to be tolerogenic, that are not properly initiating an immune response, are characterized by an increased glycolysis, glucose uptake, and lactate secretion, which also is usually considered immunosuppressive. And this is the case also in melanoma patients when you derive monocyte-derived dendritic cells from these patients compared to healthy donors. You see that there's this particular metabolism that is different and seems to be problematic. And she shows that these dendritic cells are deficient, and this deficiency kind of already stems from the monocytes that are used from the patients. Then another talk was also very interesting from Stephen Schoenberger from La Jolla, in which he was looking into adaptive cell therapy using neoantigen-specific CD4 T cells, and trying to understand whether the presence of neoantigen-specific CD4 cells is helpful because they are educating the cytotoxic T cells and the B cell responses, or whether we know that sometimes CD4 cells are known for driving direct cytolytic function. So they have this mouse model, this tumor for mice called a murine squamous cell carcinoma model that was MHC2 negative under every circumstance. Also in the presence of interferon gamma, which is a situation with some tumors upregulate MHC2 expression, and he shows that in this case, CD4s are important for mediating the response. So a proper immune response against this tumor requires both CD8 and CD4. And he shows that this is because the CD4s are the function of the CD4s, because they cannot be cytolytic because there's no MHC2 for them to respond to. They are very important for initiating or for kind of training the CD8 cells and their function in the tumor. And he also showed, he was really trying to kind of show the best practices to generate such CD4 neoantigen specific T cells. And unsurprisingly, he shows that there are certain cytokines that are better. IL-15, IL-7 versus IL-2 makes more durable CD4 cells that can generate kind of stem cell memory cells that sustain a longer, more sustained response. They show and basically if you transfer these neoantigen specific CD4 cells, the tumor can be eradicated through training of CD8 cells in a CD40 dependent mechanism. And also, he is looking for postdocs. So, Stephen Schoenberger at La Jolla. So, if you like this topic, maybe reach out to him. Then I hopped to Major Symposium D, topic aging, obesity, and adverse immune responses. But I stayed mostly for the obesity and a little bit for the aging. So, the first talk I saw was from Samir Baez. He's at Coldstream Harvard Lab. And he was talking about dietary regulation on the immune cells and stem cells in the gut. And he has already had some publication which he showed that high-fat diet affects the stem cells in the gut and can induce proliferation through a PPR delta activation and Wnt activation from PPR delta signaling. And this is induced by fatty acids in a high-fat diet. And in his previous work, he has shown that this can initiate or sustain tumorigenesis in the gut. And he proposes this as a kind of a mechanism to connect high-fat diets with increased intestinal cancer. And so in his lab at Cold Spring Harbor, he's continuing with this work with him, trying to understand how does the diet influence the immune system in surveillance and the immune characteristics of epithelial cells in the gut. And he shows that also high-fat diets downregulates MHC2 expression, which can happen in gut epithelium, and that this also has the added effect of reducing immune surveillance on these potentially cancerous cells. And he also presented kind of a comprehensive atlas. He's working on this big project in which he's feeding many mice with different types of kind of more or less defined diets and trying to understand how these different diets affect the microbiome and also the composition of the immune system after this. So if they feed them fat, if they feed them particular oils, fish, or trying to understand how diet affects these aspects of the gut immunity. And he also kind of find very interesting one particular type of nutrient, in this case, omega-6 fatty acids, which he shows that promotes regeneration of gut stem cells. He does some organoid experiments first, but then he shows also that it increases kind of recovery after damage of stem cells in the gut. And this is through arachidonic acid, which is a metabolite from these omega-6 fatty acids, and that through prostaglandin signaling, this generates epigenetic modifications that mediate stem cell plasticity and promotes regeneration and healing in the presence of arachidonic acid. Very interesting, and well, of course, work in progress.
Did he talk about omega-3s at all as well?
No, he focused on omega-6 here. His talk had a lot of data. I'm kind of picking up some of the parts, but he had a lot of different topics. His lab is working on a lot of different angles, connecting nutrition and immunity in the gut. And so then also, I think a highlight was also the presentation from Susan Cake from Salk Institute. She was looking into metabolic control of CD8 T cell exhaustion. And I thought it was really cool because she was looking into how acetyl-CoA, or like acetyls in the metabolism, can affect epigenetics. So basically, acetyl-CoA, is a cofactor that it's very important in many things in metabolism, both for a purely metabolic function of like energy generation. Acetyl-CoA is generated both from glycolysis, from the Krebs cycle, and it's used to it's burned and it's basically transformed into energy. But then also acetyl-CoA is used to acetylate histones, and this of course modifies the epigenome of a cell. And she shows that so there's different enzymes are capable of generating acetyl-CoA from different sources. On the one hand, you have the forgot the exact names of this is synthetases. One is a synthetase that generates acetyl-CoA from acetyl, and then another one that generates it from citrate. And she shows that exhausted cells have a lower proportion of one of the acetyl-CoA synthetase that makes from acetate is reduced. And she shows that having different enzymes that make acetyl-CoA from different substrates is related to the exhausted status of the T cells, and that then this affects how the acetylation of the histones happens, and this is related to either the further development of an exhausted status, or the maintenance of more of a progenitor exhausted phenotype that allows for more. So basically, effector T cells have one of these synthetases, ACSS2, and this protein is decreased in exhausted CD8 T cells, and in these cells is lyase expression is maintained, and this drives a different epigenetic program compared to the previous one, because it does not metabolize acetate as well as in the case of effector T cells that are not dysfunctional. And so exhausted cells prefer to utilize glucose for histone acetylation, whereas effector T cells rather use acetate, and that this correlates with their the kind of epigenetic and histone acetylation that they are able to do. So I think it was a very complex talk, but very interesting.
All right. That sounds like I have to stop eating gelato or something, and less fat.
Yeah, you're what you eat. That was one of the takeaway messages from those sessions.
Anyone who's done carbon labeling knows that.
Yeah, seriously, certainly. So yeah, it was a very exciting third day.
Yeah, well, you had DCs in DC.
And I tried to move beyond the T cells, so I went to see some dendritic cell of action too.
I'm very proud of you, and you even did alimentary canal work with diet, because it all acts on the gut.
Yeah, that's true. Yeah, that I can never get away from that, can I?
You're at Immunology Conference, the intestine is everywhere. All right. Well, that brings us to the episode of our latest Immunology 2023 episode here. We'll have another one tomorrow. Don't forget to follow us on Twitter at IMMUNOPodcast to find out what we're up to at the meeting, and visit us at the Immunology Podcast booth if you're here on the exhibitor floor, where you can win a prize and find out how you could be featured on a future episode of the podcast. Check back again tomorrow, and we'll be recapping another day of the Immunology 2023 conference. See you then.